All right, so it's my great pleasure to introduce Greg Stevens, who is visiting us all the way from Amsterdam. We're lucky to have him because thanks to the APS March meeting, which is happening in Chicago, and we grabbed him while we could. So Greg did his PhD in general relativity, I guess a while ago. Spent some time as postdoc in Los Alamos, I believe, before coming to Princeton, which is how I first met him. He's currently in University of Amsterdam and simultaneously somehow in Japan, which is an arrangement I have yet to figure out exactly how it works, but it sounds very cool. And he does physics of behavior. He started the eigentrend. He is the author of the original eigenworm, which was followed up by eigen. I keep seeing this in many places, like that really took off for ways of understanding. But it's all about, Greg is thinking very deeply about the emergent behaviors of, well, organisms who actually behave, and what kind of theory could we possibly have that would explain and let us think about behavior of animals. I'll stop talking there and just let Greg.
Thank you, Michael. Before we start, how long do I have you done?
You have basically the hour, so
I won't take the whole hour. So thank you for. I'm really just happy to be here, and I have some good discussions already with people. Thank you, Michael, for the introduction and for the invitation. Is it okay if I stand here? That's a reasonable spot?
yes, you are perfect.
Okay. So I'm going to talk to you about behavior of animals, so things generally with the nervous system. The title is totally pretentious and funny, right? The idea is basically we have no theory. I will just tell you the punchline at the beginning. We don't have a theory or multiple theories. And the question is really, what's the process by which we might find one? How do we think about this kind of system? And one of the things that I want to convey, in addition to the computational and analysis ideas that I'll present, is also this is a really exciting time for us thinking about physics on the scale of organisms, because it's possible to query how organisms move in a way that wasn't possible really before. And I'm not going to tell you about our work with a superorganism with a honeybee hive, but just to give you a sense of what is possible now, this is from our own sort of machine learning tracking. You can follow every bee in a hive without tags, without anything particularly complicated for quite a while. And then that's kind of your microscopic dynamics on which you would build whatever it is that bees do. I think in bees, in the worms that I'll talk about, in other systems that we're working on, this is really exciting, all facilitated by essentially the tremendous advances in machine vision, and that gives a lot of room for theorists to ask, why is the system behaving quantitatively in the way that we see and that we measure? You can absolutely interrupt, just jump in if I say anything that you want to elaborate on. You can't take bets. Well, that's interesting. Oh. Oh, there we go. That works. All right, that was late. Okay, so before I get started with the systems with the nervous system, I just kind of want to map out a little typo down here. I want to map out how this has worked in the past with, I think, a little more controllable system, and that is a system that you probably know something about. This is chemotaxis in E. coli, and it starts with beautiful imaging of what the animals are actually doing, whether it's in this case or in 3D. On the right-hand side, sort of quantification of the movement that you see. You have an idea that somehow the text got, you have an idea of what's going on here. So in this case, we know about the run and tumble model, I suspect. This comes from, molecular motors that turn a flagella for flagella clockwise or counterclockwise. Once you have an idea of that producing the movement, producing changes in the behavior in the direction of the movement, you can ask, in this case, there's no brain, but there is sort of a chemical brain, and you can go into that system and ask, as people have now for sort of a whole series of beautiful works that is ongoing, you can ask, how is it that these microscopic dynamics, these really sort of control circuit dynamics, lead all the way to the behavior and vice versa, right? And I think that intersection between the sort of fundamental control scale and the behavioral scale is something we're really interested in. It is hard to imagine how we're going to do that in more complex organisms, but we're going to give it a shot, and we're going to come from the behavioral scale down. See if all right. So let me get all my movies going. And I think I can do the fish at the same time. Okay, so I got interested in thinking about behavior in part because, similar to the ecological dynamics that Michael and others work on, it's very powerful because you feel like on that scale, you have access to the degrees of freedom that you want. And this is in contrast, for example, to most systems, let's say in neuroscience, just as an example in the brain, where what's the scale of a network that you're interested in? What is it, 100,000 for a cortical column or something? And that's really hard to measure. There are examples, including in the worm, where you can measure the whole brain. I think that's really exciting, but in general, I got interested in behavior because it's a scale that we really do have access to and at high resolution. So I'm thinking about posture. And there's another reason, which is that, depending how you count, so if we count neurons, for example, we go from none to a few hundred in the worm to 100 billion in you in between the animals. Right? So that seems like a huge explosion in complexity. And that's not associated with an explosion in the behavioral complexity. You're obviously more complicated than a bacteria. I am not insulting you, right? But I don't know if you're 100 billion times more complex than a bacteria, to be honest. So it seems like the behavioral dimensions really don't scale in the same way as kind of the internal components. And for us, that's powerful because that means that behavior is sort of we can interrogate behavior in a way that's sort of we can really sort of sample the space because it's much lower dimensional than the other ones. Okay, so that's kind of a fundamental interest in behavior, and of course we just got back from APS where we've sort of built a community and helped build a community of people working on the behavior of organisms, and in addition to all the computational advantages, they just do cool stuff. And so I should get my pointer. This is from a new experiment system that we built. These are male zebrafish. And you put male zebrafish in a tank. So we're tracking in 3D, I can tell you that offline. But you put male zebrafish in a tank after they've been isolated for a while and they fight. Presumably, some contest for dominance. It's really interesting, lots of stereotype behaviors, goes on for a long time, and then it's gone. They figure it out really fast. We don't know how it works. So by looking, by interrogating those dynamics, we hope to be able to say something about the process by which they come to this decision on who's the dominant fish. And here they're lining up for some mouth wrestling. Okay. So this is kind of the start of most of our theoretical work, or sort of more theoretical work, computational work, in part just because it's a much easier system to work with. I don't know, people in the audience know much about C. elegans, but it's one of the big genetic model systems, about 1,000 cells in total, a third of which, give or take, are brain cells, fully sort of, a pretty well understood connectome between the cells, not entirely, and then of course all kinds of powerful behavioral tools. It does, here's an example of some of the tools. These are developing now and really interesting. I won't tell you anything about this. It's from a collaborator of ours. So this is Yeah, it's okay. This is This is effectively the brain of the worm. You're measuring sort of between 100 and 200 of its neurons simultaneously. So that's really exciting. So you have the chance to bring brain and behavior together. And so there's a lot more to do in the future. And we just got really interested also because, and I really like this is a video from a longtime collaborator, Will Grew, who's now at the University of Toronto. And on the left, you're seeing high resolution imaging, microscopic imaging of the posture of the worm. On the right, you see a red square denoting where that tracking microscope is. These are about a millimeter size animals. And there's an interesting question. So there's a copper ring to keep the worms confined. And there's an interesting question, which is just, can I model those dynamics that I see, you know, in quantitative detail? And asking that question, answering that question turns out to lead us in all kinds of remarkable ways. Okay, so and here's an example, another example of different kinds of behaviors. This is typically what a biologist would look at. So here's an example of a worm in what's called a forward state. Again, these are kind of by eye states. Here's an example of a worm going backwards. It's kind of hard for people not experts to tell. And here's an example of a worm turning. And really, these are sort of canonical classes of behavior, but the worm is much richer than those classes, and that's part of the origin for some of our work. So, Michael mentioned eigenworms, which is rather embarrassing because really it's computationally trivial. But the idea was simply, and the idea extends, that what you really want when you think about behavior, high resolution behavior, is you want not just where I am in the room, you want what I'm doing. There's a lot of information in the way that I hold my posture. Same with the worms, same with the fish, same with all the things that we look at. So what does the posture of a worm mean? You get an image like this. It's pretty high resolution and very clean. You can extract the centerline curve, a centerline curve, this approximation of this shape. And then what we mean by the posture of this animal is simply the geometry of that centerline curve. And once we've abstracted this image to a centerline geometry, here this actually corresponds exactly to this image, then you can collect many, many of those images that's just coming from your tracking microscope. And you can ask simple questions like, what is the space of postures that during this experiment, the animal's actually exploring? And the answer turns out to be pretty much 4 for the worm. We can quibble about 5 or 6. All right, and how do we know that? We know that because we basically just do singular value decomposition on this kind of data. And we get eigenvalues that are, these are the cumulative eigenvalues. By the time we get to 4 or 5, basically we've gotten everything, okay? And so this is kind of what I mean. It means that I can take a worm and a worm shape and I can decompose it into these 4 basis shapes with time-varying amplitudes, A1 to A4. They're very interpretable. Doesn't have to be the case. They're very interpretable so that A1 and A2 effectively capture a wave that's moving up and down the body. They're out of phase with each other, so it defines a phase variable. And A3, the third one, is a turning mode. So when this one is excited, you get, in positive, you get these large shapes, and negative, you get these large shapes. And I'm going kind of fast over this because I want to get to the real stuff for today, but just to give you a sense, oops, I was supposed to be, there we go. So here's, I think, what we want for many organisms, sort of at least unconstrained behavior on the right, our preliminary analysis in the low-dimensional shape on the left. And what you saw in red was when we zap the worm with a heat impulse, it's going to do a different behavior in response to that. In fact, what's going on now is an omega turn, and you can follow that. We can follow that trajectory in some detail and ask about what's going on in those space of trajectories, okay? And there's hints, of course, that even this is all just statics, right? It's analysis of posture, but there's already hints that these are directly related to behavior. Of course, in this joint distribution, anytime you find a circle in your data, it's a good thing, right? So already there's hints of dynamics here and maybe over here and here where we see sort of particular shapes, okay? But nothing particularly fundamental. Okay, we want to get at behavior, which is the dynamics of posture. But we have no equations. Absolutely no one knows how to write down an equation for the posture of the worm that's rich enough to capture the movies that I saw. And so here's an example of a pendulum where we do have equations, but we don't have those for the worm. So the question is, you can think about the pendulum as maybe some of you in the audience already have. Instead of thinking about the equations of motion for the pendulum, you can recover the dynamics of what's going on by looking at the trajectories in the phase space, right? And that's going to be the approach that we take. So we're going to reconstruct the phase space. So in this case, I can tell you that it's really simple harmonic down here, and then if I give it enough energy, it's going to go sort of around and around and around. So can we do something similar for the worm, and what do we learn by doing so? Okay, and really the idea is to quantify the dynamics that we're getting from the microscope. Oops. Okay. So this is kind of a terrible graphic, but it does convey the point, and I think it's a fundamental point, and let me explain a little bit. The idea is when you measure something, could be the worm, could be something else, you don't really know the system. You don't know whether the measurements that you're making are enough, right? And there's theory for dynamical systems anyway, and the idealized case where there's no noise or little noise, that if I only measure, let's say, the position coordinate of a dynamical system, I wasn't measuring the velocity, but I took lagged measurements of the position. So I took not just the position at time t, but the position time t plus 1, t plus 2 eventually, then I could recover the dynamics of that system in those lag coordinates. This is a well-known idea in dynamical systems, but we think it's sort of fundamentally important to querying complex living systems because who knows where the dynamics actually sits. So we built sort of a data-friendly technique in which we could sort of add these lagged values of the posture modes and ultimately infer sort of a lower dimensional, hopefully lower dimensional space in which those dynamics actually exist. Okay, and if you do this right, at least so the theory says, so here's your original dynamic, sorry, that's your actual underlying system. You're making some measurement of that system. You try to reconstruct that system through this what's called delay embedding, sort of capture short time predictability. And at the end of the day, you get a candidate system, this one, that you hope is identical to the system that you actually started with, that you didn't know about. And there's theorems about when that can be true and so on. And I think the most important thing I want to convey is that there are often many ways of, well, first of all, that you should think about your time series as having information well beyond just the instantaneous measurement. And if you can think about sort of bringing in that near instantaneous measurement in a principled way, it can be very powerful. Okay, so we built this for the worm. Here's how it works. So you're concatenating postures in time. How long you go, we have a measure of predictability. So we're adding in frames until we get kind of a local maximum in how predictive the system actually is. I'll talk more about that as we go on. It turns out to be about half a second or so in the case of this sampling of the worm. So we add that in. We look at the loaded, so this is k times 5-dimensional space, so k star times 5-dimensional space. We do an SVD within that space and find ultimately that we need 1, 2, 3, 4, 5, 7 of these. So this M star is now sort of measuring the dimensionality of the inferred, not the configuration space, but the inferred phase space, the inferred state space. Okay? And I'll come back to how we do K and M a little bit later in a different context. It will be, I think, a little clearer. And what do we find in the worm? So remember that we had these posture modes, these static configurations that told me sort of the instantaneous posture. These are now short snippets of dynamics There are two of them that describe a wave going down the body. That's gamma, it's gamma 1 and gamma 2, or gamma forward 1, gamma forward 2. There are two of these that describe a wave going the opposite direction, which is why the slope is opposite. That's a reversal dynamics. And there are three of these modes, the last three that make up the 7-dimensional space, that describe turning. All right, and there really is a difference between the static posture modes and these dynamics, just like there would be if there was an underlying velocity and you were only measuring position, right? So the state space and the posture space don't have to be the same dimensionality simply because we're taking into account dynamics.
Sorry.
Yes.
So in the pictures that you're showing, are there 12 like K-star rows in those pictures? They look more than that.
There are 11, I believe. There are 11 delays of a system that was 5-dimensional to begin with. So we took 5 of the posture modes. So it's 5 times 11 as the in principle full dimensionality of state. 55.
But the pictures you're showing there, how many rows are there? That's separate. That's like
Where?
like the pictures that you're showing to illustrate the modes.
No, it's the same. I haven't shown you the actual dimensions.
But there, each row is presumably already the sum over your 5 eigenmodes. That's the actual posture.
Now, each one of these is now, now we're doing SVD within this space. So it's going to be a filter over those 55 dimensions, which I'm not showing you.
Okay.
Make sense? So it will be a little simpler, I think, later, but think of it this way. Imagine you just have a really simple particle and you're measuring it. It's a Newtonian particle, so you need the velocity and you need the position and the acceleration, or position and the velocity to evolve things forward. So then K star would probably be 2. In the no noise limit, it would be exactly 2. Why? Because you need that second frame to define the velocity. That's all. Okay, so that's really all that's going on here. There's a mixture with noise and so on, which is why it can be a little hard to know a priori what K star M star should actually be here.
I guess my question is just that it looks like those images are much smoother than if they had 12 rows. So that's what I'm confused. And K stars or 11.
This is a 55-dimensional thing. Right? And I'm not showing you the actual filters for it. That's all. Yeah. We'll come back. Maybe we can go on and come back. Okay. So what do these things look like? Again, just to get some intuition. So if you look at the two forward modes and measure them against the velocity, just the centroid velocity, which can be positive or negative, then in the forward state, those are always positive. In the reverse state, they're negative, and it looks like the forward state and the reversal state are quite different. And then this is what it looks like in the turn state. And then just to, again, to add some interpretability, coming back to that situation where we zap the worm on the head. So then it does something called an escape response. It goes through a series of behaviors from reversal to turn, and then back to forward crawling, and just ask, what's the projection onto each one of these subspaces. So the stimulus happens at time 0. It goes very quickly into the reversal subspace. It diminishes the forward crawling subspace, which is green. And then as the reversal ends, it starts to turn, and ultimately it comes back to what it was doing a little bit before the stimulus. Okay, so that's what they mean.
Sorry, can you explain the first figure? Is it like XF1 and XF2, and the trajectory there? That's so a warm. Sorry, I'm lost.
Oh, so what's going on there?
Yeah.
So these are short. These are these short snippets, and as you move along in time, these change values, and that's all that we're showing here. Just projecting the 7-dimensional space onto this 2-dimensional space of these modes. and you're just color coding them by the average velocity in that at that time.
Velocity in that space, or
No,
velocity,
the velocity as in the centroid velocity, that just the position velocity that you have, as this kind of a separate measurement, just to connect the two.
Okay, and so these dynamics here, the trajectory of the worm is in 7-dimensional space, which is something you projected out of your 55 thing.
Yeah.
Okay,
Yeah.
and here you're projecting it onto just this
That's right.
two.
That's right. Good. Make sense?
So this worm is the trajectory is in a plane.
The worm, yes. So the worm is crawling on an agar plate. They can lift their head up a little bit, but usually don't do that so much in the conditions that we've arranged. But they do live in 3D and move in 3D when you kind of set the conditions so that they can. And there are other people who are trying to study that in some sort of agar matrix or something. But for us, it's 2D. Yeah, good.
There's some intuition for why the reverse modes seem to explore larger regimes than the forward.
Yeah, so these are just much more unstable. Actually, it looks like an unstable spiral. Is that what you mean?
Yeah.
Yeah, and I think
Okay.
that's just a part of the control system. It doesn't really stay in the reversal state for very long. Exactly. Yeah. Okay. Other questions? All right, we'll go on a little bit. Okay, so just a higher resolution movie of what I showed you before. The idea is now going to be what we want to say something about this behavior through these modes, through this reconstructed state space that we built, the 7-dimensional thing that we think is capturing what's going on here, right? And I just showed you those different projections. So then you have to think about, well, I have these variables. What do I do with them, right? You could fit equations of motion if you wanted to, but I don't think that's particularly valuable. One of the nice lessons from dynamical systems is there are certain invariants that you can try to measure, right? Which would be independent of the way we parameterize or build coordinates on the state space. We're going to try to measure those invariants and see what they tell us about worm behavior. Okay, so one of those invariants for certain kinds of systems, especially for chaotic systems, is especially in a system like this when you seem to have a lot of cycles. So again, this is real data, but projected into a particular perspective on the 7-dimensional space. And there's a bunch of cycles in here that they don't seem to be exactly on top of each other. So you can go into the 7-dimensional space and effectively just identify and count the orbits that you see. All right. Yeah.
For some sort of sense of scale, do you know how much, if there's some noise in fitting, what's the scale of noise on this picture? For if the next time I traverse the cycle, it's a little bit off. Is it the worm that's doing the same thing slightly differently, or?
Right. So I'm going to try to argue that it's the worm that's doing something slightly differently, and it's quite challenging to know what the scale of whatever. We don't know what the scale, if there is noise, what that scale could be. So we're going to try to argue that by quantifying the variability that we see in this cycle-to-cycle behavior, that it's structured in a way that makes it very unlikely that it's just noise. That's going to
presumably there's some naive intuition, at least from the kind of I've heard Gordon give his talks and some setting up some naive intuition about how the behavior might work. Presumably there's some level of intuition where it's people might expect some cycle and maybe I bump into another one and there's some other cycle, and this seems to be a much simpler system and it's completely already not true.
Yeah, there isn't that intuition, at least not that I know of. Yeah. So let me try to show you what the variability looks like, and then we'll come back and see if you're convinced. Okay. So we identify cycles. So you look, and they could be of varying periods, and these are all the cycles that we find. We measure along the cycle how stable the cycle is, which is just looking at the Jacobian along those cycles. and they're all unstable.
So how do you define a cycle?
You define a cycle as a recurrence in your state space to some value epsilon. So it carries with it a metric distance, which is kind of how close do you consider a recurrence to be. And try to be careful about that. And that's what a cycle is. Okay, yeah.
Are these cycles like replicates from recordings, or are they like each of them a slightly distinct behavioral posture? Like if you have like many things that are like kind of overriding themselves in the picture, are those?
So here's an example where you have these, this is a longer cycle, and these are different, but very close. Right? You would actually say that those are the same, those are the same cycle revisited later. Right? So it's like the skeleton of orbits, and so this is the same, it's not happening at the same time, but it's the same orbit, as if you came back to the same orbit and did it again. So
So you would label them as two different cycles?
we would label them as, no, we would label them as the same orbit, this topological structure of an orbit, and then we would say that you revisit that orbit sort of multiple times in the course of your dynamics. So the picture that we're trying to build here is that there's, you know, in the structure of the reconstructed state space, there are these cycles. You can be quantitative about what those cycles do for you, and that is that, so not only are they all unstable, but if you ask, so I don't know if people have heard about a Lyapunov exponent or a maximum Lyapunov exponent. You can just measure that by looking at local divergence in your trajectories. If your system is really tiled by these cycles, you can also measure it by the distribution of the cycles. And if those two agree, then that gives you support that these are one and the same thing, and that's what we're showing here. So where these unstable orbits, which are these orbits that I'm talking about here, sort of if you take the spectrum of those orbits and describe the system in terms of where you are in those orbits, then you reproduce actually I didn't show you on the previous slide, the maximum Lyapunov exponent of this system, which is a nice sort of, by no means trivial alignment between the pictures. And this was the example I was telling you about, where this is an example of a cycle that just revisited multiple times in different times, or even by different orbs. Actually, I'm not sure what the case is here. And we're not the only ones. This is basically dynamical systems. This is chaos in dynamical systems. In fact, having a spectrum of unstable orbits is equivalent to chaos, more or less. All right. And you know, the challenge is, of course, how can you be sure when it's from data? And I just want to throw out that we're not the only ones who are thinking about this. These are hard objects to define because they're extended in the state space, but there was recent work doing this in fluids, right? Where, you know, you can describe turbulence as bouncing from one of these, now it's a spatial temporal orbit, bouncing from one of these orbits to another, okay? And it seems like, just to point out that it's not just us, okay. This might be a little more interpretable, or maybe not. So another, or actually kind of a fundamental invariant from which you can construct most everything else that you need, is a measure of basically the Jacobians averaged across the called the Lyapunov spectrum. And what are we measuring? So we're taking a ball around our trajectory. We're asking, as the trajectory advances, what happened? This ball is the same number of dimensions of the state space to begin with, right? And we're asking what's happening to the dimensions of that ball. There are 7 of them. Some of them are shrinking. Some of them are growing, maybe. And they could all be shrinking or staying constant. What's going on with that ball? That's measured invariantly by the Lyapunov spectrum. That spectrum is characterized by 7 exponents, and that's what we're measuring. The distribution comes from some bootstrap sampling. So these are the 7 exponents. There are 2 chaotic exponents, so 2 exponents that would drive exponential divergence. There's 1 near 0 exponent, or 0 exponent, which would be the case if it was a continuous dynamical system. You expect on general grounds to have a 0 exponent. and there are 3 negative exponents such that the overall sort of volume of the state space is contracting. It's a dissipative dynamical system. Okay, and so this would, this would align, these 2 exponents would then align with this idea that there's this instability bouncing from one orbit to another. The structure here is what I mean. This is not something that is in any obvious way created by noise. It has even more structure if you fold the spectrum among this particular point here. It's symmetric, right? And just to give you a little bit, some intuition about what that means, if you had just a double pendulum, a Hamiltonian chaotic system, right, which would have 4 degrees of freedom, the 2 angles and their 2 coordinates and their 2 momenta, right? Then you would have 1 positive exponent, 1 negative exponent of exactly the same magnitude and 2 zeros. All right, so a Hamiltonian system is purely symmetric. Right? So what we're seeing here, we think, is an indication of a nearly Hamiltonian system presumably broken by dissipation. Right? So a Hamiltonian system somewhere inside whatever's controlling the biomechanics of the worm, driven and damped. Yep.
Sorry. 2 questions. One is, are there 6 exponents there or 7?
7.
Where's the 7th?
There are 6. Where's the 7th? Oh, it's here. It's just not shown. There's always an sorry. There's an exponent right on here. I just didn't show it. Yep.
And yeah, this thing about it being symmetric for Hamiltonian systems, maybe it should be obvious to me, but why is it not just that they sum to 0? That's like the sort of Liouville theorem.
It's symmetric because it's time reversal invariant. So if you have a positive exponent because of chaos, then you have to have a corresponding negative exponent, and they have to be exactly the same, so that there's no breaking of time invariance, time reversal invariance.
And if you just like by hand added, I don't know if there's an analog of adding noise to computation, like you have some data and you can imagine that there was extra noise.
You mean, could you somehow get this from noise?
Or like, can you see that noise destroys that? Because you said that this structure in a way tells you that this is not just noise.
Right, so noise, if you started with your double pendulum and added noise, you can destroy this, because then it's not really a continuous system. And so you can destroy this. We didn't sort of explore around too much more than that. Okay. Let me give you a sense of why I don't think it's crazy at all. And that is that ultimately this is coming, the behavior of the worm and behavior of other organisms, including this Drosophila larva, driven by, at least in part, by the biomechanics of the body. So what we think actually is going on is there's a relatively simple neural controller somehow, right? An effective controller coming from the nervous system. And then there's kind of elastic deformations of the body, of which we see kind of some hint of that already, all right? And that would be powerful in a way, because that would mean it kind of fits this idea about how much you offload from, let's say, your controller to your body mechanics. One of the things that is very important that you might want to do is explore. When you're going to explore an environment, you have to roll the dice somehow to move around, right? Otherwise, how are you going to explore? So maybe there's a noise source somewhere, right? Or maybe you just use the sort of natural variability that you get from the kind of body mechanics that make you up, that makes up the body of the worm, and that's what we think is actually happening. We're very actively trying to build a reasonable biomechanical model that explains the spectrum. Yeah.
So if I take that logic to an extreme, if you use your heat pulse experiments, would you recover these same states and these same exponents, and you just sort of reconfigure which ones you use? Or?
Great question. So you have to also be able to get stereotypy in some behaviors, and the heat pulse is a great example, because this is part of an escape response that worms do. Well, in that case, in response to heat, but also in response to measuring fungus. And the structure stays the same, but you squeeze the overall variability. If I built this out of data that was only associated around the heat pulse, you would see the spectrum still have the same structure, but shrink. So we think, and that just shrinks the overall variability of the response. So that we think is under control of the worm. Exactly. And this brings me to my first point, which is that there's no way, I'll back up. There really isn't an easy way from these videos to know what an interesting theoretical question is. I mean, I could tell you that you should model the behavior, but I can't tell you much more than that. I mean, we could talk about the statistics of the trajectories and so on, but on the posture scale, there's no obvious way. We have to do some work. And so the theory reimagined part of the title is you have to do some work bringing all the tools that we know before there's a place that we're going to make contact with a deep theoretical question. And we think this kind of structure is a deep theoretical question, right? Not just, and we don't think it's just about the worm. I think there's other systems for which this is equally true. Okay.
So the structure as an example of the question is like,
now,
how does it become more stereotypical in like stress response?
No, well, first of all, just where does this come from in a much more controllable way, in a model, basically. Now you know now you have a sense of what a goal of the model can be. Does that make sense?
Is there something to grasp upon, like what could be a stupid null model that people have tried, maybe, or one could try? Like what is easy to reproduce and what is very hard?
You know, so one model for behavior, which I don't think is viable, is that a behavioral state is like a limit cycle. Like you get stuck in it. But, you know, that's not a good idea, right? Because you're stuck and you're not very flexible. And although that idea has been around in biomechanics for a while, we don't really see any good evidence for it. So this does not seem to be a collection of stable cycles. That's off the table now, right? And in at least the way we see it.
And like the limit cycle, another limit cycle, and some sort of control.
It's not limit cycle versus, it's stable versus unstable. So this would just say that you're never stuck in a particular trajectory. You're bouncing all the time. So it's a picture of behavior in which, you know, stereotypy is a little bit challenging here because yes, if you go back to, you know, a picture like this, okay, if I look from a distance, you know, those are cycles. But every time the worm, you know, basically every cycle, the worm is doing something a little bit different. And so it's not really a limit cycle, it's a collection of cycles. And it's that, and you're bound, you know, it's that collection both in reverse and so on. And you're just sort of bouncing around from all of those. And that's not really a picture that has been, so that would be our contrasted picture with limit cycles of behavior.
And again, we know that that's not just, you know, the data analysis, image processing procedure that kind of returns these slightly different values because of that structure and the spectrum somehow.
There's just no obvious way to get this combination of sort of to see this, the fact that the cycles reproduce the exponent, the fact that you get this kind of Lyapunov, this kind of near symmetric spectrum. Maybe there's a way. I'm not, it's data driven. So that's why I think the model will make it strong. I didn't mention that in not our work, but in other work, people have already sort of observed this kind of idea in Drosophila larvae. It's basically a biomechanical model that explores the world through Hamiltonian chaos. So it's not crazy, but you know, the real connection remains to be made. Okay, all right. We're gonna jump. Well, Michael saw this already. It may be, oh, I can't do this because I don't have any sound. So I'm just gonna skip him. The second, so the first part was on variability. So let me just emphasize, we built the dynamical system from data precisely to query where is the variability coming from, or what can we say about the structure of the variability, which seemed to be a useful question and now led us to kind of a direction for modeling motor control. Another interesting question, is there are longer lived dynamics in behavior, as in many systems in biology. And how would we query a system if we didn't know a priori what they should be? And so let me give you something that you'll probably remember. So you're used to thinking, you've thought a lot about Langevin dynamics, simple Langevin dynamics for whatever system you're looking at. So stochastic trajectories, but there's an alternative formulation, which is to not think about the trajectories like this, but to think about ensembles of trajectories. This is the Fokker-Planck equation. What's the difference? They're very similar, right? Except that the big difference for us, the very powerful difference, is that fundamentally the Fokker-Planck dynamics are linear. Even though, so everything that I just described is what you have to do when you have sort of nonlinear dynamics. You're querying a system that has nonlinear dynamics. That's powerful but tricky. The stuff about orbits is topologically, it's an extended object. So we wanted a way to encapsulate the dynamics that would allow us to say some things about the behavior and also be computationally tractable. And this Fokker-Planck perspective seems reasonable. And just to remind you how it works. So this is our particle in a double well. It's an example I'll say a lot about. It's bouncing back and forth. Finite temperature, there's enough thermal fluctuations to bounce it across the well. And depending on the temperature, these transitions could be extremely rare, in which case you wouldn't necessarily obviously see the kind of long time dynamics, that is the transition rate that you expect to see. And also there's a lot of noise, so looking at these trajectories short time can be very challenging. On the other hand, if I look at the density, in this case just a position, I start all the particles in the left well, and this density just evolves monotonically through the Fokker-Planck equation until it reaches its equilibrium state at long times. Okay? And this dynamics is encoded by this linear operator here, which you can expand as we like to do in eigenfunctions and eigenvalues. And the eigenvalues of that Fokker-Planck operator tell us about long time scales in the system. So in this case, there's always a unit. Well, depends a little bit about whether you do this discrete or continuous. Let's talk about discrete. There's always a unit eigenvalue in the discrete case. That's for the probability sort of summing. And then in this case, there's one extra non-trivial eigenvalue, longer-lived eigenvalue. That's simply the hopping rate, which you could see in the spectrum. And then there's a bunch of other things. Okay, so in the formalism that we already have some experience with, sort of this Fokker-Planck versus Langevin formalism, is an idea that maybe we could identify long time scales from the data directly, and like the spectrum, like the Lyapunov spectrum, if there is a long time scale that you can identify quantitatively, then you have a question of where it comes from and how to model it. That's the rationale. All right. And the fun thing is you can do this from data. You don't need an equation of motion. You don't need the Fokker-Planck equation. You can learn the Fokker-Planck dynamics from data. So we revisit the sort of state space reconstruction that we did before, but in a slightly different way. So we have some system, we sample it with k delays. So it's a multidimensional system, but we expand our state in time with k of these embeddings, k of these delays, and we partition the system, sort of put little boxes on the phase space, and we partition the system as finely as we can. So we expand the complexity of the dynamics until we have a fine enough partition that we measure by the entropy. So a maximum entropy partition here at n star. This kind of puts a fundamental bound on the size of our phase space boxes. And once we have that, then we use that partition dynamics to measure the entropy and ask when does the sort of local entropy, this Markov entropy, when does it stop changing as a function of k?
Sorry, is n the number of
And
boxes?
n is the number of boxes, a
And
number
what-
of partitions.
And what limits, like what makes the entropy go down? And it's the entropy of,
That's totally finite size effect.
okay, it's the entropy of what? It's the entropy of the
It's the
distribution.
Markov entropy of the time series that you're measuring. So for any value of k, you have a state, and that state gets split into n phase-space cells, and you just measure the one-step Markov probability between those state space cells. You use the entropy of that Markov process, and that's what we're measuring in the vertical age.
So at some point, if I introduce a billion of them, then it just in my data, I didn't have enough samples. So every time I saw this, which was exactly once, next time I was there, which was also happened exactly once. And so the entropy is 0 again. Is that how it works?
You mean if you keep going over
Yeah,
here?
just to understand like that.
And so this gives us, and why is this powerful? It's maybe a little technical, but the idea is that what we're really measuring is this is predictive information through a Markov process. So assuming the process is Markov, we are putting a state space partition and building these number of delays in a way that's sort of maximally predictive. So it's a way of building your best Markov model out of the dynamics that you have.
But isn't there a massive question? How do you pick, like, let's say you decided that you're going to go with 10 boxes. How do you draw the boxes?
Oh, we're doing this on k-means based on the data that we actually have. So there is absolutely freedom in how you partition the space. Yeah. So doing this in a way that respects the invariance of a continuous dynamical system is a challenge, but totally doable. Right, so you could ask, and in fact, we didn't get a chance to talk yet about it, but this is turning our effectively continuous dynamics into a discrete process. That's generally a bad idea. So you need to be able to show me that this discrete process of your continuous dynamics, like a Lorentz system, actually respects the continuous dynamics that you started with, and at least toy examples, you can do that. Right, and again, it's one of those things where often people may not think twice about how you partition your, certainly when it comes to C. elegans behavior, it's going forward, that's one state, it's going backward, that's one state. All we're saying here is we can be more careful about building that partition. It's a very fine partition, and then we're going to do something with this partition state space. Okay, what are we going to do? Now, I want to get I can take 5 minutes. I'm going to get the key idea across, and then I'll summarize. The key idea is really simple. Once you have this partition state space, the dynamics, the Fokker-Planck dynamics that you're interested in is simply the transitions from one cell to another. Right? So all you have to do is count. Right? And so that's going from this continuous dynamics to a finite time, still continuous dynamics, to approximating that finite time dynamics with basically just the transition matrix between state space cells, reconstructed state space cells. This matrix is now encoding all the dynamics of your reconstructed state space. And that matrix, it's a linear, this is just a linear operator, so it's the eigenvalues and eigenvectors of that matrix that have something interesting to say, or could have something interesting to say about the dynamics. And in particular, this P will always have an eigenvalue 1. That's the Perron-Frobenius theorem. Again, it's just the idea that probabilities, this matrix is a stochastic matrix, right? So it's either row or column normalized, but not both in general. So we'll always have an eigenvalue 1. If there's if there's a long time scale in the system, there will be an eigenvalue that is close to 1. And that's it, in the sense. So if we can find that, and we can do something with the eigenvector associated to the eigenvalue that's long-lived. So if you want a way to query the data, it tells you whether there's a long time scale in the process, then this seems like a useful way, okay? And here's the idea that in our rich reconstructed state space, here's sort of complicated graphic of fluids or brains or whatever, then maybe there was a long time scale, and that long time, if I look at the eigenfunction associated with that eigenvalue, then I can draw a partition and talk about the dynamics corresponding. So that's effectively learning a dynamics that's like a metastable dynamics on this state space. Okay, so it works. So here's a simple example of the double well where it's transitioning back and forth between the two wells. I'm not going to dwell on it. This is what happens when you partition that space. You get ultimately, in this case, it's fundamentally a stochastic system, so the entropy never truncates, so it would always keep going, right? But you can do the embedding, and it was an underdamped particle, so essentially you need at least one more delay. That's k. This is k equals 2. You need more than 2 for reasons that are fundamentally interesting, but a little technical about what happens to stochastic differential equations. I'm going to skip that. Okay, and this will help with the intuition. So this is what's happening to the probability density when we start in one well and we look at how that probability density is evolving. The time scale for that evolution is set by the second eigenvalue. If I learn, again, through this transition matrix, I learn the spectrum. In this case, I'm writing not the eigenvalues, but an implied time scale, which is just given by this transformation. Then there is one eigenvalue close to one long time scale. That long time scale is the hopping back and forth between the wells. So it's learning this coarse-grained metastable dynamics on top of your system. And again, if there isn't a timescale separation, it is really not obvious, by the way, when you start with the timescales of the postures, that there's any kind of timescale separation, right? But if there is one, then this would be a way to see it. And just do the punchline. So this is what it looks like for the worm, doing the whole process, blah, blah, blah. And at the end of the day, it's not nearly as well separated as it is for the double well. There's one longer time scale. And there's a whole bunch of others. This is not unexpected for a living system. But that longer time scale is really interesting. I'm just looking at the spectrum of the inferred transition matrix here. That long time scale is really interesting. It's effectively the run and tumble model of C. elegans. So called run and pirouette. It means that you go for a quasi-straight, you're doing this for a while, and then you start turning, right? So this is what a trajectory looks like. And these points, the blue and the yellow, are labeled by the eigenfunction, right? Not by us. And the timescale separation is not great. So these, typically there's we're talking about a dwell time of, sort of 23 seconds in the run state, 3 seconds in the turn state. So it's not huge, but it's certainly there, and it's totally biologically relevant. So people came up with this name well before we apply this operator, and it pops out very nicely in this operator formula. Okay, so again, the point related to the beginning of the talk is simply this gives us a way to query our system. Where else are long time scales interesting? Maybe you think there's a system for which there's no obvious order parameter. So we study, say, groups of fish interacting with each other, not large groups, but groups of 5 or 6. How would you even write an order parameter for such a system? Or how could you learn about an order parameter from the dynamics? Order parameters are usually slow, right? So finding slow dynamics can give you a big hint as to what's going on in the system. Okay, so I'm going to skip that part. Just going to add this. So, we're bringing in the tools that you all know already. So dynamical systems, which maybe physicists should know a little bit more about. Statistical physics, information theory, those are all common. But we're doing this, like I said, in a region where there isn't really good models or good theory. So then you have to think about, well, how do I build a dynamical system if I don't have the equations of motion? That's here. how do I query the Fokker-Planck equation if I don't have it? And of course, the ideas of entropy information theory are just sort of threaded throughout. But we really do need to think about doing this in this kind of inverse way. So with that, Michael mentioned that we have a group in Amsterdam and a group in Japan. I don't know how that works either, but it does work. And if you're interested in anything, we'd love to have you visit when travel is easy again. So thank you for your patience.
So because of the sound thing, our Zoom participants, if you want to type anything in the chat, I'll voice it, but.
Okay. Great. Yeah.
So I'm curious how we think about stimuli-driven attractors or state switching, I guess, more generally. So either in the case early in your talk when you had these unstable libraries of orbits, or later when you had these like red and blue colored metastable states, how do we think about stimuli inducing changes between those, or is that not really fitting in with what you've done so far?
No, it's a great question. And we think about the,
Repeat the question just because I don't know if that microphone picks up the question.
oh, I should repeat the question?
If you could,
Okay,
yeah.
yes, good. So the question is how to think about stimuli in this context of orbits or in the context of the long time scales. And that's a great question. And what we think happens and what we see in the worm is when this impulse arrives, you kind of exert, you push your trajectory out of the space that we normally see, but you get onto one of these unstable orbits that's different than where you were before. So essentially you get onto a reversal unstable orbit or an escape response unstable, and then you let this sort of passive dynamics take over. So it would imply that, so it's totally within the framework that we've talked about, but it would focus the control on the early part of the stimulus, and then you let the dynamics take over again. So you push yourself to the right part of phase space, let yourself go again, and that would be that stimulus response. For the kind of long time scale, this long-lived dynamics, I don't know yet, but I think, so the eigenfunctions, the modes associated with the long-lived dynamics are the things where you would naturally include a stimulus. And I think you'd naturally write down some sort of driven sort of equation for those and should be fine.
Or like the collection of all the orbits that are even like the forward, there's many different forward orbits from my understanding, and they're all unstable.
Mm-hmm.
So do we think of that as like you're relatively likely or easy to kick yourself from one forward to another, but it's like some threshold that is harder to get from a forward to a reversal?
Great. So the question was, if we have a whole collection of unstable forward orbits, it's relatively easy to move between those, harder to move between orbits coming from reversals or turns. The interesting thing is just hopping from one, just letting the system naturally move from one orbit to another reproduces kind of the natural diffusion in the phase space that you see. So you don't need any active process for the exploration of the phase space. It just happens by being there.
Question. So is there a sense in which, so for your, I guess, 7-dimensional space, so you're showing some projections, but kind of your interpretation of this as a dynamical system before we turn on the stimulus, kind of, am I right that it's kind of like it's a dynamical system at every point I have a vector pointing somewhere, but the idea would be that if I happen to come back exactly there, the vector would be pointing in the same direction. Like in this space, the lines don't cross.
Correct.
Like they get a diversion because of instability, but they wouldn't actually cross. Is there a sensible way? Obviously, there's everything. There's with a plus or minus epsilon, and there's some finite. Is there some sensible way in which you test if it's true for this interpretation? Like, have you? Because if it's obviously violated by the data, that means there's something else you didn't take into account.
Right,
The
right.
worm is happy and then unhappy, and that somehow just determines where it turns.
Right, so you can try to measure that directly. So should I repeat the question?
Oh, that was so long.
So there's a way to ask in your, so the question was in, are there crossings in your reconstructed state?
And how do you know you included
that would
everything?
tell you that you kind of have incomplete information. So there's a way to query that directly, which is actually the way we first started. Called one of the ways is false nearest neighbors. So you can, and really what that means is that you look at your neighbors at one dimension, and if they're false, they go away as you add embedding dimension, right? Because they're actually not close, right? So you look at points that when you add embedding dimensions, they move away really, and you count how many of those.
And the points are what here, and the dimensions are what?
Sorry, the points are whatever space you have, right? And you ask whether there's 2 points in the, I have a space coming from some embedding process, and I measure the distance between 2 points in that space. I look at those same 2 points in an embedding process that has an additional dimension, and I ask whether they moved apart or not. If they move apart by a lot, that's probably because you were under embedded. Make sense? And so you count those as a way of counting how many crossing, how many approximate crossings do you think are, and that gives you, and that also aligns with the 7. So that's one way to ask, at, so kind of at the limits of the resolution, whether you're really getting crossings or not. I should say, in one other interesting fact, which I always forget to mention, these are all done on worms off food when they're exploring. You can put worms on food. And in fact, some of the videos, maybe I think I showed you, have worms on food. And it's remarkably more complicated because they're sitting around, there's food, they look much less stereotyped. But in fact, you reproduce the exact same state space plus 8. So 8 more dimensions for eating. And it's not crazy. They're a lot more complicated. So it totally sort of separates. So you get the same, you reproduce the crawling, this non-food crawling state space, and then you add additional dimensions corresponding presumably to being on food and all the stimuli associated with being on food. And you can get rid of all those 8 with a particular gene perturbation, which desensitizes the worm to the mechanical feeling of bacteria. And if you do that, then you're back to the 7-dimensional space. It's all quite, it feels solid, right? Like that's something I wouldn't, so it's not so tied to the particular data that you use. I thought that was kind of fun. That was pretty.
Wow, I have more questions, but probably best over coffee or dinner. Let's thank our speaker again. There's a question from Anders. Sorry, I have to voice it just appeared. If this is a Hamiltonian system, do you have a rough idea of what the Hamiltonian is?
Stay tuned, because that's a great question. And so we're building based on kind of relatively straightforward elastic continuum models. We're building the Hamiltonian. And we have a pretty good idea about what it should look like. So stay tuned.
Awesome. There's Anders is there. We can hear you probably.
Oh, okay. You can hear me. Thank you. Yeah. Never mind. I'm going to turn.
No, that's my fault. I was hoping we would hear him. He said thank you.
No, it's a great question, and I think it will be really nice to have this model in our hands.
I guess we'll have to have you over again. Tell us what the experience. Well, thank you very much so much. Let's. There's more coffee. There's still cookies.